Today, I would like to talk about the severe pandemic situation of the present world. In the last century, the world witnessed three major pandemics. That's Spanish flu in 1918, which is caused by the influenza virus, that's H1N1, is considered as the mother of all pandemics, which affected 1/3 of the world population and nearly 50 to 100 million deaths were reported. In 1957, Asian flu and in 1968, Hong Kong flu were the other two pandemics. Severe acute respiratory syndrome coronavirus, that's SARS CoV, and Middle East respiratory syndrome coronavirus, that's MERS-CoV, are the two highly transmissible and pathogenic viruses that emerged in humans at the beginning of the 21st century. The outbreak of COVID-19 has become a clinical threat to the general population as well as healthcare workers worldwide. If a severe pandemic materializes, all of the society could pay a heavy price for decades, especially in the healthcare system and the economy. Nature has its own rules and follows an unknown and sometimes violent design that cannot be predicted. We all know that the novel coronavirus disease is highly transmittable and pathogenic viral infection which is caused by SARS CoV2 virus. The intermediate source of the origin of the virus is not known and rapid human-to-human transmission is confirmed widely. Moreover, this virus is highly contagious than influenza and fortunately less than that of measles. Coronavirus disease was first described in 1931, but the first coronavirus HCOV229E isolated from humans in 1965 only. Since there is no treatment for COVID-19, the best way to prevent the illness is just avoid being exposed to the virus. The SARS-CoV-2 or COVID-19 is a single-stranded RNA-positive virus which belongs to the genus beta coronavirus within the family Coronavidae. This virus is thought to spread mainly from person to person between people who are in close contact with one another. That's up to 7 feet and maybe through microdroplets produced when an infected person coughs, sneezes or talks loudly. These microdroplets of size 5 to 10 microns can penetrate the human body, that's lungs, via inhalation through the nose or mouth, and that's why social distancing is more important in preventing the infection. Coronavirus is an M protein enveloped virus with spike proteins S1 and S2. There is also a lipid protective bilayer on the outer surface of it, which is actually derived from the host cell membrane itself. This spike protein helps the virus in attaching the host cell membrane. Fortunately, only 20 seconds hand wash with soap help us to destroy the viral lipid bilayer because soap can dissolve both water-soluble as well as lipid-soluble substances. 70% alcohol-based sanitizer is also a good lipid solvent and hence can destroy the viral lipid layer. If we can damage the spike proteins or the lipid bilayer, then there is no scope for the virus to attack the host cell membrane. Humidity, UV radiation and temperature are the other factors affecting the stability of the virus outside the body. Clinically, there are three stages of SARS-CoV-2 infection. The first one is an asymptomatic incubation period with or without the detectable virus. The second stage is non-severe symptomatic period with the presence of the virus, and the third stage is severe respiratory symptomatic stage with high viral load. The stage 1 individuals being healthy carriers also can spread the virus unknowingly. And we all know that the viruses are non-living entities and do not inherently have their own metabolism. The binding of the virus with the host cell receptors is a significant determinant for the pathogenesis of infection. The coronavirus S protein has been reported as an important tool for virus entry into the host cells. Among functions of structural proteins, the envelope has a crucial role in virus pathogenicity as it promotes viral assembly and release. The envelope spike glycoprotein binds to its cellular receptors, that is angiotensin converting enzyme 2 for SARS-CoV as well as SARS-CoV-2. EC2 could be expressed in the oral cavity and was highly enriched in epithelial cells, tongue, buccal and gingival tissues. Remember, that's why the mucus of oral cavity may be a potential high-risk route for COVID-19 infection. High EC2 expression tissues identified in the body are alveolar cells of lung, esophagus, stratified epithelial cells, obstructive enterocytes from ileum and colon, cholangiocytes, myocardial cells, renal proximal convoluted tubule cells, and urothelial cells. Hence, these organs with high ACE2 expression cells are considered as potential high risk for infection and organ failure. Many researchers are focusing to prevent the attachment of the virus by manipulating ACE2 expression in these cells. Immune responses to viral infections are a complex interplay between the virus, target cells and the cells of the immune system. Virus-specific CD8 cytotoxic T lymphocytes, which can eradicate the virus by direct and indirect mechanisms. And dendritic cells are the most potent antigen-presenting cells and play a central role to activate cytotoxic lymphocyte. The entry of SARS-CoV-2 into the dendritic cells may be mediated through C-type glycans such as CD209 and not by ACE2. Dendritic cells capture antigens and induces self-maturation and migrate to secondary lymphoid organs where they can activate the naive T cells. Activation of naive CD8 T cells and polarization towards effective cytotoxic lymphocytes require presentation of MHC class I peptide complexes together with co-stimulation and the presence of cytokines such as interleukin-12 and interferon alpha. Viral antigen presentation subsequently stimulates the body's humoral as well as cellular immunity, which are mediated by virus-specific B and T cells. Similar to common acute viral infections, the antibody profile against SARS-CoV virus has been typical pattern of IgM and IgG production. The SARS-specific IgM antibodies disappear at the end of 12th week, while the IgG antibody can last for a long time, which indicates IgG antibody might play a protective role in it. The latest report shows that the number of CD4 and CD8 T cells in the peripheral blood of SARS-CoV-2 infected patients is significantly reduced. Recent research underlines that nuclear shuttle proteins is able to block the host's innate immune response. The acute respiratory distress syndrome is the main cause of death of COVID-19. Out of the 41 SARS-CoV-2 infected patients admitted in the early stages of the outbreak, 6 died from ARDS only. ARDS is the common immunopathological event for SARS-CoV-2, SARS-CoV and MERS-CoV infections. One of the main mechanisms for ARDS is the cytokine storm, which is the deadly uncontrolled systemic inflammatory response resulting from the release of large amounts of pro-inflammatory cytokines. This cytokine storm will trigger a violent attack by the immune system to the body, which cause ARDS and multiple organ failure, and finally lead to death in severe cases of SARS-CoV-2 infection. No drugs or biologics have been proven to be effective for the prevention or the treatment of COVID-19. Numerous antiviral agents, immunotherapies and vaccines are being investigated as potential therapies. So we have to strictly follow the social distancing, wearing masks and frequent hand washing with soap or sanitizer. Moreover, everyone should be vigilant to increase the natural immunity by adding immunomodulators and antioxidants to the daily food. Investigating rare diseases gives researchers more clue about how healthy immune system functions. As Albert Einstein said, we still do not know 1,001% of what nature has revealed to us. The mutations undergone by the coronavirus are the biggest challenges to the researchers around the world. At least 30 variants of the virus have been noticed by the scientists, and this becomes a hurdle in developing a new drug against this dangerous virus. Nothing in life is to be feared. It is only to be understood. Now it is time to understand more so that we may fear less. So we will break the chain with the scientific knowledge.